Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Monday Seminar Series. Before I introduce today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. I encourage you to add your questions as they arise during the presentation. And each month for this academic year, our semester series has featured a speaker from one of the USDA Agricultural Research Service Human Nutrition Research Centers. And today's speaker, Dr. Nancy Moran, is an assistant professor at the USDA ARS Children's Nutrition Research Center at Baylor College of Medicine in Houston. Dr. Moran received her bachelor's in molecular and cellular biology with a minor in chemistry and her doctorate in nutrition sciences from the University of Illinois, Urbana-Champaign. She completed her postdoctoral training in Steve Clinton's lab at Ohio State University. Dr. Moran's research focuses on the role of carotenoids in supporting health across the lifespan, as well as the role of carotenoids as biomarkers of dietary intake. And she is the incoming chair of the Gordon Research Conference on Carotenoids and the past chair of the American Society for Nutrition's Carotenoid and Vitamin A Research Interest Group. And today, Dr. Moran will discuss her carotenoid research in a presentation titled Advances in Assessing Fruit, Vegetable, and Carotenoid Intake in Pediatric and Adult Populations. And with that, let me turn the microphone over to Dr. Moran.
Okay, thank you so much for that nice introduction. I appreciate it. So we'll go ahead and get started talking about some of our research we've been able to make progress in for the past couple of years on how to utilize carotenoid biomarkers for assessing fruit, vegetable, and carotenoid intake in a variety of populations. Okay, I have no conflicts of interest to disclose, and an outline of what I'll be talking about today is here. First, we'll do a little review of where do we stand in the United States on fruit and vegetable intake in our different age groups. Followed by that, we'll talk about the potential for carotenoids to serve as biomarkers of fruit and vegetable intake in different age groups, and then some of the research that I along with my collaborators, have been able to do to define the validity of skin carotenoid biomarkers in adults, adolescents, toddlers, and infants. And we'll have time for questions at the end. Okay, so where do we stand on fruit and vegetable intake in the United States? So the CDC morbidity and mortality weekly report has lots of good information available on this. So first I'll start with this panel on the left side here, where they published in February 2023 that among young children aged 1 to 5 years of age, 1 in 2 children do not eat a vegetable daily. And 1 in 3 children do not get a fruit daily. So you can imagine there's a lot of kids out there that aren't touching a fruit or a vegetable at all in a given day. Now, what about when we look at older kids and adults? In US adults, the median fruit intake is 1 serving per day, and the median vegetable intake is about 1.5 servings per day. Of those adults, 12% of our adults meet fruit recommendations and 10% meet vegetable intake recommendations. When we looked at slightly younger people, our adolescent population, we find a big drop in their fruit and vegetable intake, with only 7% of adult adolescents meeting fruit recommendations and only 2% meeting vegetable recommendations. So the point of this slide is to tell you, even though we know how important fruits and vegetables are in supporting a healthy diet and meeting those essential requirements for healthy living across age groups, a lot of Americans, regardless of age, just don't meet those intake requirements. We have a lot of work to do with developing better interventions to support better fruit and vegetable intake. And in that manner, we need to actually be able to track fruit and vegetable intake better. Okay, so in comes carotenoids in the diet. So I'm going to pivot here pretty quickly. So what are carotenoids? So carotenoids are actually fat-soluble plant pigments that are involved in light harvesting, antioxidant protection, signaling, and attractants in the actual plant tissue. Carotenoids are not produced by animals. In order to be obtained, they have to be obtained from the diet, and this is common across all animals, from sea sponges up to humans. Some carotenoids are converted to vitamin A in the body. So, for instance, beta carotene, which has these terminal unsubstituted beta ionone rings here, which you see, can be cleaved centrally by an enzyme called beta carotene oxygenase 1 to yield all trans retinal. This is our first form of vitamin A that's generated, and then that can be metabolized to retinol, which is a circulating form of vitamin A. Then that can be metabolized to generate retinoic acid, which many of you would know is a critical regulator of gene expression and is the ligand for retinoic acid receptor. Okay, so in addition to serving as vitamin A, other carotenoids exert a variety of other functions, some of which you would have learned from other seminars in this talk, perhaps given by Dr. Wong. And they also act as antioxidants in general, owing to this long chain of conjugated double bonds. Okay, so I'm telling you what carotenoids do in the diet, but then what we're going to talk about later is that also as a function of their color and their presence across many, but not all fruits and vegetables, they can also serve as a biomarker of fruit and vegetable intake. But before we get into that, let's talk a little bit about how we absorb and distribute carotenoids from those foods. So since carotenoids are fat soluble, they're absorbed and distributed similar to other fat soluble compounds in the diet. So as a refresher for you, so carotenoids are released from the food matrix with chewing and with digestion. Then carotenoids are mixed with lipids and emulsified in the intestine. Gastrointestinal enzymes help those to form mixed lipid micelles, and those mixed lipid micelles interact with proteins on the surface of absorptive enterocytes to be absorbed in the small intestine predominantly. From there, carotenoids are packaged into lipoprotein particles, either chylomicrons or HDL particles through reverse cholesterol transport pathways, and are then distributed throughout the body, generally first to the liver and then to the periphery. Many carotenoids are stored in the liver as a depot of provitamin A, but then can also be metabolized and utilized there, as well as distributed to peripheral tissues, including skin, which is the main tissue we'll be talking about today in terms of carotenoid biomarkers. All right, generally, what do we know about carotenoids and health? Because I do want to impress before we switch to the carotenoid biomarker angle of the talk. I just want to talk generally about, we know a lot about the role of carotenoids in adult and a little less in pediatric health. So what do we know in adults? I'm just going to summarize here. There's a lot more references that we could give here, and I encourage you to check these out if you're interested. Carotenoid intake from foods and plasma carotenoid concentrations are generally inversely associated with the risk of several cancers, including prostate, lung, breast, and head and neck cancer. Cardiovascular disease risk is also inversely associated with plasma concentrations of beta carotene and lycopene. Visual function across the life cycle is associated with carotenoid intake, owing partly to the accumulation of carotenoids in the macula of the eye, and can confer reduced age-related macular degeneration, glare reduction, and improved acuity and contrast sensitivity across age groups. And then last but not least, there's increasing research, some of which has been done at your institution, on the relationship between carotenoids and improved learning memory across age groups from the very, very young infants up to older people. Okay, now we know a little less about the role of carotenoids and health in pediatric nutrition, but I do want to share some of the knowns and unknowns for this age group. So like I said, we know that carotenoids are provitamin A compounds and so can contribute to the vitamin A requirements for healthy growth and development. However, I would argue that the infant requirement for vitamin A is not super well defined, so we can make progress there. We know that infants obtain carotenoids during gestation in utero and then after birth via breast milk. There's no recommended intakes of carotenoids for any group, though. We do know that adults meet about 30% of their total vitamin A requirement from provitamin A carotenoids. We don't know specifically what the importance of carotenoids is for meeting vitamin A requirements in children and during pregnancy and lactation. Like I said, we know in older adults that carotenoids are related to cognitive and visual function, and there is some recent research on that effect in elementary age school kids as well as infants. Carotenoids are also associated with reduced oxidative stress and are anti-inflammatory, and that was shown in a study of infants who were supplemented with carotenoid-containing formula. And then last but not least, one of the things, since I'm really interested in young children, we also know that there's just large changes in carotenoid intake across childhood. So there's definitely carotenoid intake coming from milk, and then as kids start consuming pureed vegetables, then their carotenoid intake really shoots up, and then that might kind of come down a little bit as they start switching to a more typical older kid diet. Okay, so I told you what we know kind of about the role of carotenoids in adult and children's health. But what about our American intake of carotenoids? So here's what it looks like right now. So adults are shown in the green bars and children are shown in the blue bars. The major carotenoid consumed in the United States is lycopene, and that's the red carotenoid in tomatoes. Americans love their tomatoes and tomato products. Followed by beta carotene, which is found in orange and dark green leafy vegetables, then lutein and zeaxanthin predominant in yellow vegetables and also dark green leafies, then alpha carotene and beta cryptoxanthin. And then I have on here a comparison with the intake of preformed retinol and then total vitamin A intake when you combine sources of provitamin A carotenoids with preformed vitamin A. And I will just point out that kids consume a lot of lycopene that's similar to the adults, but otherwise you see that kids consume less of the different carotenoid species than adults. All right. Our lab, just to understand what is the relevance of carotenoids during the infant exclusive milk feeding stage of life, did a systematic review of all of the publications on carotenoid content in milk that we could find, and we published those results in Nutrition Reviews in 2022. So I did just want to share this, that we found that certainly colostrum, which is the earliest milk known for its golden color, has the greatest concentration of carotenoids compared to the different milk stages. And then that decreases during the transitional milk lactation stage and then is lower, but definitely present in mature milk. So carotenoids are definitely present in all stages of human milk. And this is reflected in newborns and exclusively milk-fed infants. So when I mentioned that during gestation, carotenoids are transferred to the infant, and that's reflected in newborns where they are born with an existing pool of carotenoids. And we broke those categories up just into the US and non-US just to see if there were any big differences. Then you see that as their diet starts to diversify and as they've had time to consume carotenoids from milk and formula, their blood carotenoid concentrations increase. And then those blood carotenoid concentrations further increase after 6 months of age when kids really start to incorporate solid foods and some of these bright, colorful, carotenoid-rich vegetable purees. That's definitely reflected here in their blood carotenoid concentrations. So I've shown you that carotenoids are related to health across ages, and that people in the United States of all ages consume some amount of carotenoids, even if it's not as much as it could be given the relatively low vegetable intake. So let's talk about research that we've done to validate carotenoid intake assessment tools in children and adults, and we'll probably start with the adults here. So why are we interested in this issue? So objective nutrition assessment in humans is really difficult, but it's so important for our field of nutritional sciences. Everyone knows that using diet assessment tools for different populations can be challenging, because to get a really accurate assessment of what a population eats creates significant participant burden and or significant researcher burden. Other quick screeners and tools sometimes are just too quick to be precise also. So when we think about assessing carotenoid or just fruit and vegetable intake in children, we run into additional issues, which is for children, we would usually go to their adult proxy to report on their carotenoid intake, but it's hard to find a single informed proxy for a child given how many caregivers children are with in a given day. Some water. Like I mentioned, survey tools can be time-consuming or too brief to be accurate, and blood biomarker measures are great, but those can actually create a participation barrier depending on the age group and the research setting that you're working with. Some people, it just might be difficult to even pull off getting a blood sample and then getting those samples analyzed in the lab. So the implications of these complexities are that if we don't know what children and adults eat, how can we actually relate their diet to nutrition and health? Or how can we determine whether or not our nutrition and policy interventions are actually working or not, or if they need additional tweaks? So an area that our lab has been looking into has been developing carotenoid concentration biomarkers of dietary intake using reflection spectroscopy specifically. So plasma carotenoid concentrations are the current standard objective biomarker, I would say, of fruit and vegetable intake, and those can be measured by high pressure liquid chromatography or LC mass spectrometry. Skin carotenoids provide a kind of an alternative to that carotenoid exposure assessment. And skin carotenoids can be measured by either Raman spectroscopy using like a custom device that's shown up here in this image, or reflection spectroscopy can be done. And there's a number of different commercial reflection spectroscopy devices. The Currently, the field is moving a little more toward the reflection spectroscopy devices because these are commercially available in research grade, whereas the Raman devices are typically, if they're out there, they're not considered research grade, or if you want a research grade device, those need to be a custom build because they're more expensive because of the laser technology that has to go into these. On the flip side, reflection spectroscopy operates particularly in this device called the Vegimeter using a white LED and then a filtered detector. So the technology is less expensive, making it more amenable for commercialization. So my talk is really going to focus on validation of this reflection spectroscopy tool that's found in the United States called the Vegimeter. So what's the premise of how this tool works? So like I said, it uses a white LED. It can also use a tungsten or halogen bulb to illuminate the skin. Then the light of the chromophores in the skin is reflected back into the device and then detected with a spectrograph. And then that data is analyzed and displayed for the researcher, and it's also recorded on the accompanying laptop device. So that's how the device works. Now, what's the illumination or spectral window that it's measuring? So that is a spectral window of 460 to 500 nanometers, and that's demonstrated here kind of lightly on this figure. And let me walk you through this. So along the y-axis is the absorption of different compounds, and then the wavelength in nanometers along the x-axis. And so what the makers of the device have done is they've shown a sketch of the absorption of different major chromophores in the skin. So here, this line, this solid black line, represents the absorption of carotenoids. The lighter line represents the absorption of oxygenated hemoglobin. The dot dash is unoxygenated hemoglobin, and then kind of this other dotted line is flavonoids. So the principle here, and the melanin is not shown in this, but it's very similar absorption to the flavonoids. So the principle here is that they're measuring in a window where there's less interference coming in from hemoglobin, from flavonoids, and from melanin. But then they further improve the performance of the device by building the device with a lever, shown right here, that applies pressure to the finger to temporarily blanch the finger, pushing away blood, and then further minimizing this hemoglobin interference at the carotenoid spectral window. So this technology, I'll show you, it looks like it's performing pretty well in adults, but we have been interested in just also validating this in different age groups because skin thicknesses can differ across age groups as well as their diets. So to that point, we'll be covering the validity of reflection spectroscopy-based skin carotenoid biomarkers in adults, adolescents, and infants and toddlers. And then I'll introduce a new study funded by NICHD to validate the skin carotenoid biomarkers in infants and toddlers using a larger study design. Okay, so when I talk about biomarker validity, what exactly do I mean? So there's a great article out by Dragstead that really walks through the different components of validity of a biomarker. And so I found this to be a very helpful framework that when we're thinking about validating a biomarker, there's different things we're looking for. So we're looking for plausibility. Is the measurement of what you're looking at for the biomarker actually related to what you think you're measuring. So in our case, carotenoids would plausibly be biomarkers for carotenoid-rich foods like fruits and vegetables. We also want to show that there's a dose response effect. If you increase your intake of carotenoids, does it actually increase the signal strength of the biomarker? Is there a predictable time response? Do you have a sense of how long is it going to take from the time that that food is eaten to the time that the biomarker changes? Is it robust in a variety of populations? So key things we've been focusing on have been different age groups, and then also American racial groups and ethnic groups to account for differences in skin melanin, as well as differences in dietary patterns that we might see in the groups that we work with. Also, is the device reliable? And then is there analytical stability and performance reproducibility? So first, I'm going to talk about some of our research in adult populations on these three components: dose response, time response, and robustness in a variety of adult populations. So first, I want to share a really hallmark, very well done paper by Lisa Jans, published by in American Journal of Clinical Nutrition in 2014. So she really did a very careful validation of resonance Raman spectroscopy, not the reflection spectroscopy, but the resonance Raman spectroscopy, showing that total RRS intensity, so the signal of skin carotenoids, was related to changes in the diet, and that those changes were in parallel largely with plasma carotenoid changes. So in this study, participants had a baseline measurement followed by phase 1, which was a washout. Then in phase 2, they for 8 weeks consumed 6 servings of fruits and vegetables from pack out and dine-in meals, which equated to 60 milligrams of total carotenoids per day. Then they went on to a phase 3 washout diet, and then they returned to their own diet in phase 4. So I think that this study shows very nicely that this is plausible, that carotenoids coming from increased fruit and vegetable intake lead to an increase not only in plasma carotenoids, but also in skin carotenoids over the course of 8 weeks. From this study, she also established that the skin carotenoid intensity in these 29 adults was positively correlated with the plasma carotenoid concentrations. So this was just a wonderfully done controlled feeding study, but we really wanted to pick up from this and establish whether the same thing would be true in reflection spectroscopy, and also in a more diverse and larger cohort of adults. So this study being done in North Dakota was predominantly European ancestry participants. So we wanted to expand that. So along with my collaborators, Stephanie Gilcott-Pitts and Melissa Laska, we conducted both a cross-sectional and a controlled feeding study. So first, starting with the cross-sectional study, we enrolled participants in Minnesota and in North Carolina. There were 213 participants enrolled in total, generally more women or females than men, and then a distribution of self-categorized race ethnicity groups. The average age was 34 years of age, plus minus 12 years, and then pretty healthy BMI. So what we found when we looked at the correlation with the reflection spectroscopy skin carotenoid score, we found that total plasma carotenoids were significantly correlated with skin carotenoid score with an R of 0.7. We found that skin carotenoid score was positively associated with fruit and vegetable intake with an R of 0.35, and positively correlated with self-reported carotenoid intake with an R of 0.38. So with that, we wanted to move on to a controlled feeding study to take a closer look at the dose response and time response. So for this study, we enrolled participants again in Minnesota, North Carolina, and then at my location in Houston, Texas. We were able to enroll 162 participants, and they consumed either 0 milligrams of carotenoids per day for 6 weeks from an apple juice product, 4 milligrams of carotenoids per day from an orange carrot juice product, or 8 milligrams of carotenoids per day from the same orange carrot juice product from 12 ounces of it. And these carotenoid intakes are kind of representative to adding 1 to 2 servings of vegetables per day. It depends on which vegetable you're looking at, but that's kind of the way we conceptualized it, is that these aren't huge amounts of carotenoids. We did baseline measurements, midpoint measurements, and end of study measurements, and had good retention in the study. We tracked intervention compliance, measured blood carotenoid concentrations, anthropometrics. At each visit, we also did measure skin melanin to determine whether or not that was a significant covariate for the changes in skin carotenoids. And then we did an assessment of baseline diet, sun, and tobacco exposure, which we'll get back to later in the presentation. Here's a carotenoid profile of the juice that we provided. Since it was a carrot-based juice, it is predominantly beta-carotene and alpha-carotene rich, with low amounts of the other carotenoids. So I think that's important because there's a possibility that different carotenoids would behave differently. Okay, so here's the results that we found. So here's the distribution of participants, fairly evenly distributed between the 3 sites. Intervention compliance was very high, 96 to 90% of the 8% of the target dose was consumed by each of the groups, and we had 98% retention of the participants in the study. We evenly distributed participants across summer and fall, with a lower number enrolling in the final season of winter and spring. And then there were even enrollment of males and females, and this study enrolled even percentages of different self-categorized race ethnicities. The education and income levels are shown here, and then the mean age was 32 years of age, and again, the mean BMI was 25. Their starting skin carotenoid score was 298. Okay, so what did we see over time? So this figure is going to show time along the x-axis, and then raw skin carotenoid score at each visit for the 3 groups. So solid bar is the control group, so you can see that stayed pretty flat across the study. The dotted line is the moderate dose, or 4 milligrams per day, and the dashed line is the 8 milligrams per day. So you can see that there was a significant increase in skin carotenoid score units. And then when we adjusted for the baseline skin carotenoid score, we saw that there was indeed a significant mean separation. So we could discriminate between the 3 groups based on the change in skin carotenoid score. I do like to point out how this contrasts with plasma carotenoids using both the raw data and the baseline corrected data. So we'll focus over here on the right, but here's the uncorrected data for your reference. This change in skin carotenoid score, you see it just keeps going up, 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 up. There's no plateau in this 6 weeks. So if we wanted to find out how long it would have taken to reach equilibrium, we probably would have had to keep going quite some time, because this didn't really show much of a sign of slowing. On the other hand, you can argue that for the moderate group, there was a plateau at 3 weeks, or we met some kind of equilibrium for plasma carotenoids. So what does this mean? So I guess it means that the plasma carotenoids, as expected, are a faster responding biomarker of intake, whereas skin carotenoids might be a marker more of longer term carotenoid intake patterns. But I think more work is needed for us to figure out what is that time frame that's reflected with skin carotenoids. So our strengths in that study are that we were happy to have diverse U.S. adult participation in different geographic locations, excellent compliance, and nutritionally relevant doses. But the limitations were we didn't reach that plateau in the skin carotenoids, and we did have to use a juice matrix in order to really control the intake well instead of intact foods, and as a consequence of that, the juice was enriched in just 2 carotenoid species and maybe not all the carotenoid species you would see in the US diet. But there's really an opportunity here for future studies to investigate time, food matrix, and carotenoid species effects as determinants of skin carotenoids. Okay, so that's some of the really rigorous work that we've been able to do in adults. So now I'm going to show some of the work we've been able to do with younger populations. So like I said, we think this is important to validate this biomarker across age groups for a number of reasons. So first of all, there's just a huge change in dietary patterns in early life. From 0 to 4 or 6 months of age, infants consume one food, milk. And then that changes radically with the introduction of these first veggies and fruits, which are really some of the major foods that kids receive in that first year if they're receiving purees. Then after that, a lot of people start transitioning to different foods, foods that are easy to hold for self-feeding, and then over maybe that first 3 to 5 years of life, their diet really starts to look more like the rest of their family's diet. But even then, like I showed you, then it changes again. That fruit and vegetable intake is pretty good when they're a little younger, and then it really starts to fall off into adolescence. So we have a lot of changes and different intake ranges that we need to establish whether or not we can detect food intake within that range that's observed in the population. And then, of course, there's a lot of changes in skin physiology and body composition that happen as well, which affects pharmacokinetics of everything, including carotenoids. Okay, so in order to get into this, we will kind of work backwards from adults. So our next group down is adolescents. So we were able to work with Katie Arlinghouse, who's at University of Minnesota, and examine some data for a secondary analysis on teens that participated in a healthy physical activity and nutrition intervention at their school. So for this study, there were 171 kids that enrolled in a nutrition and physical activity program. They participated in that for the first semester of school, and then they were randomized for the second semester based then they were randomized in December to either an escalated program that had greater intensity on areas where they needed additional support, or a maintenance program that just kind of maintained what they had been doing for the first semester. So fortunately, she was able to measure skin carotenoids and also dietary intake using a shorter screener at each of these major data collection time points, as well as their BMI percentile. So what we found is here. So here's who participated. So the average age was 13 years. This was a predominantly Hispanic and African American group of students. their grade was predominantly that middle school age range. And then there were about half of the kids were a healthy weight, and then the other half were overweight or had obesity. And then as far as based on their dietary questionnaire, what was their fruit and vegetable intake for times per day? They reported 3 times per day for fruits and vegetables, and then 1.5 times per day for the fruit and 1.8 times per day for the vegetable. And their starting skin carotenoids were lower than what we saw previously in adults at 228. All right, so we wanted to know if their skin carotenoid scores correlated with what they were telling us, at least for the fruit and vegetable intake, as a preliminary look at this age group. And so we did find actually that their skin carotenoid scores were significantly associated with total fruit and vegetable intake. And this was by testing this relationship in a generalized linear mix model that was corrected for BMI, Z-score, age, sex, and time point of the study. Okay, but then what happens when we look separately at fruit? We find actually that the skin carotenoid score is not related to their self-reported fruit intake, but is more strongly related to their vegetable intake. And then if we break it down to look at their self-reported intake of orange and green vegetables, it's also strongly related to the orange and green vegetable intake. So why is this? I think it's because in the United States, the major fruits that we consume are not carotenoid rich. The top fruits are apples, bananas, grapes. Oranges have a little carotenoid, but I think that's the reason here why we don't really see an independent relationship between skin carotenoids and fruit intake. Okay, so that's what we saw in adolescence. Previously, I had found in infants that their skin carotenoid score was positively associated with their total carotenoid intake per day. And this is down to, you'll see this carotenoid intake, we're looking in a range of basically 0 to 0.5 milligram per day. So we are able to see a relationship here between skin carotenoids and carotenoid intake. And then similarly, within 4-month-olds, same group of kids, we see skin carotenoid score is significantly and positively associated with plasma total carotenoid concentrations. So at this age group where they're exclusively milk fed, this relationship looks pretty strong. But what about toddlers? And I think this is about our last group, and then I'll talk about some other covariates. We did a similar study of skin carotenoid score relationships with fruit and vegetable intake in toddlers. And again, we were able to leverage some data for a secondary data analysis collected by Tracy Ledoux at University of Houston. And she had toddlers who were participating in a playgroup-based health education intervention. That was the test intervention, and the control intervention was classroom-based health education for parents. This was a 10-week intervention, and so she measured skin carotenoids at the beginning of the intervention and at the end, and then also assessed fruit and vegetable intake using the Kid Bites food frequency questionnaire. So what we found for who participated is here. So the predominantly mothers participated as the adults. The parents were on average about 30 years old, predominantly Hispanic and non-Hispanic African American participants. And then going down to the toddlers were on average 22 months of age, and a little more males than females here. And as far as their fruit and vegetable intake, their baseline skin carotenoids was 253 skin carotenoid score units. Oops. Okay, so what did we find? We found that in toddlers, skin carotenoid score was positively predicted by total carotenoids in a model that adjusted for age, sex, BMI percentile, treatment group, and time point. And then we found that skin carotenoids were positively associated with beta carotene intake and lutein intake, but not intake of the other carotenoids. And then, like in the adolescents, we found that skin carotenoids were not predicted by fruit intake, but were strongly predicted by total vegetable intake. And then when you combine fruit and vegetable intake, it's still predicted by the sum of fruit and vegetable intake. And so each of these outcomes is coming from a different model. And then last on this study, we found that the change in skin carotenoids from baseline of the study to the end of the study was also related to the kids' change in reported carotenoid intake. So I feel like that is a useful tool for folks who are monitoring changes in diet across a time frame. So, like I said, we have a time and dose response here, but we definitely would like to move this toward being a more precise and quantitative biomarker. Okay, so moving on to the very last part, there's always a lot of questions about how reliable the Vegimeter or reflection spectroscopy biomarker is. And whether or not it's affected by variables like age, sex, season, body composition, skin melanin, skin hemoglobin. So we actually just published a paper looking at these individual variables. So I'm just going to tell you what all we tested here, and then share kind of the conclusion here. So basically, we found that geographic location was not a significant predictor of the skin carotenoid score responses for the moderate dose or high dose group. And this is based on the controlled juice study we did in adults. We also found that the changes in skin carotenoid score did not differ by self-categorized race, ethnicity, but did differ based on a person's BMI. So greater BMI was, did not differ by BMI, but did differ by starting skin carotenoid score. So the higher the skin carotenoid score was at the beginning of the study, the smaller the change in skin carotenoid score to the different juice interventions. No difference by season, sex, age, skin melanin, other skin tone components, which are measured in a color scale here. No difference by cholesterol, glucose, sun exposure. So with that, I'll say we've kind of learned that this skin carotenoid score, at least in the adults, based on this study, is robust to a number of variables. It's really just affected by what the starting skin carotenoid score is. So looking at change or looking within an individual or correcting for baseline is going to be important when interpreting skin carotenoid scores. And we've also learned that there's similar positive relationships between dietary carotenoid and fruit and vegetable intake in different age groups, with there being a stronger relationship with the vegetable intake than with fruit intake. And so based on everything I've told you, this is a study we have underway right now to try to look at a number of key different stages in infancy and early toddlerhood just to validate the veggie meter. And so we're calling this the BIT-Bonds study, Baylor Infant and Toddler Biomarker of Nutrition study. We're enrolling infants and they start the study at 4 months of age, and we're assessing their diet prior to each visit and then looking at the relationship between dietary carotenoids, blood carotenoids, and the skin carotenoid biomarker at each of the following time points: 4 months of age, when they would be exclusively milk fed; 6 months of age, when they're just starting to get those first foods; 8 months of age, when they're probably more into that early infant dietary pattern; and then 12, 18, and 24 months of age when they're really starting to transition more to table foods and family foods. So that study is underway, and that's supported by NICHD. And with that, I'll conclude and say that skin carotenoid biomarkers of carotenoid and vegetable intake can present an opportunity for group diet assessment in children and adults. And future research, though, is still needed on the validity of skin carotenoids as a biomarker of infant carotenoid intake across feeding stages. And I'll put in my shameless plug here for the Gordon Research Conference on carotenoids, apocarotenoids, and vitamin A biology that's going to be in January 2025 in Ventura Beach, California. Everyone's welcome. Registration is now open. And of course, we would love to support underrepresented minorities in science. There is a Carl Storm travel fellowship program that's open, first come, first serve, to support underrepresented grad students, postdocs, faculty, and scientists. So we encourage application to that as well. With that, I'd like to thank all of my collaborators, and I'd be happy to take any questions.
Thank you very much, Dr. Moran. I'm going to take my prerogative as host to ask the first question.
Okay.
When you're showing the spectrum, the wavelengths for the carotenoids using the reflection spectroscope, you show the wavelengths also for flavonoids. Now, has anybody tried to use this to assess flavonoid status? And would that, do you think that would be actually maybe a solution to some of the issues with the fruit intake?
I have not. Maybe somebody else in the audience might know. I have not personally seen this technology being used for flavonoids. I know that this veggie meter device we're looking at is tuned for the carotenoids. It does seem like you could potentially tune it to a different spectral window that would absorb better for flavonoids. So yeah, it seems like that could be an opportunity to pick up the fruits. Good point.
Okay, thank you. The next question comes from Larissa Constantino, who asked, do you have any idea which skin carotenoid score there would be a plateau, and where exactly in the skin do carotenoids accumulate?
Good question. Yeah, so I know this device measures up to 800. Once in a while, we'll see somebody that has a very high skin carotenoid score. But yeah, I have that same question about where it would plateau. Now, one thing that we do know is that there's this condition called carotenemia that you see in babies, often when they first start eating those carrot purees and their nose turns orange. So I think there is a potential for there to be pretty high skin carotenoids. And if you think about the amount of beta carotene that a baby is getting if they eat 3 little jars of baby carrots a day, for instance, that's a lot relative to their body weight. So it might be that the skin has a great potential to accumulate carotenoids based on that. And then where exactly in the skin do carotenoids accumulate? I used to know the answer to that question, and there is a great review written by, I think, Helmut Sees on carotenoids in the skin. And that, I think, has more information to answer that. It's certainly in higher, you can detect it in higher levels of the stratum corneum, because that's where the device is measuring. So the device is measuring, I think, resonance Raman measures about 200 microns deep, and I think that reflection spectroscopy measures to a similar depth, if not a little deeper, to up to 700 microns deep.
Okay, the next question comes from Shandong Wong, and very long question, so bear with me. Okay, very nice talk, he says. Thank you. It was great to have this non-invasive way to measure carotenoids accumulation in the skin, which may particularly help assessment of long-term carotenoid intake for individual people with tissue accumulation. We did see a huge difference between carotenoid accumulation, yellow phenotype of tissue of BCO1, BCO2 knockout mice and their plasma levels of carotenoids with much less change. I have a question. Because the reflection spectroscopy measures total carotenoids and cannot separate out individual carotenoids, I was wondering if you could use another non-invasive way, such as flicker photometry, to assess macular pigment optical density, i.e. only lutein and zeaxanthine, and see if there is any correlation between levels of skin carotenoids and macular carotenoids in your current or future studies.
Yeah, thank you, Shang, for those great comments. Yeah, and very interesting. So Dr. Wong is talking about it's called a psychobehavioral measurement of macular carotenoids. Your ability to see a flickering light in a device called a heterochromatic flicker photometer is dependent on the amount of lutein in the back of your eye. So actually, we are looking at that relationship between macular carotenoids and skin carotenoids in our pregnant population. I didn't talk about that study today, but that's something that we are working on. Unfortunately, we can't do that measurement in little kids because it requires understanding when to push a button based on their visual stimuli. And it's kind of a difficult test even for grown-ups to do. There has been some work on the validation of the relationship between eye carotenoids and skin carotenoids by other groups, I think including the Bernstein group and the Stringham groups, I think. And they find that it doesn't always correlate, I think, because the binding kinetics of the proteins involved in holding on to lutein in the back of the eye are different from the binding kinetics of skin carotenoids. So your macular carotenoids are more kind of stable and less responsive to diet than skin carotenoids from what I've seen. But there's always a possibility if somebody's starting low for eye carotenoids for them to improve.
Okay, the next question is from Saidas, who again says, nice talk and thanks you. Are there any concerns and caveats that you foresee for studies using the Vegimeter and solid foods rather than concentrated liquid foods?
Okay, so when we're talking about concentrated liquid foods, I'm gonna assume we're thinking about like an Ensure product or something like that, like a adult nutritional or maybe an infant nutritional. So I'll say if that's kind of where the question is coming from, yeah, that's a big consideration for me for using the veggie meter to assess infant diet, because the bioavailability and the exposure to carotenoids, either from human milk or from supplemented infant formula, is probably quite different than the bioavailability and exposure to carotenoids coming from vegetables or an intact food. So I think that that is something we are hoping to understand better, at least with our infant and toddler study. And so we're trying to do some in-depth assessment to see how complicated this will be to sort out.
We have one question remaining, but we've reached 1 o'clock. Do you have time for one more question?
Sure. Yeah.
Okay,
Thank
great.
you.
That's a
Mhm.
very good question. One that I was interested in myself. So again, this is from Megan Farady. Great presentation. Thank you. Do you know if the veggie meter has been tried in older adults? Older adults lose fat on their skins with age, and I'm wondering if this device would be a good method for infants and younger adults, or if it does work in older adults.
That is a great question. It may have been done in older adults, but now that I'm thinking about it, that study is not popping up to my mind. So somebody's welcome to chime in if they know of a study of the veggie meter being validated in older adults. But yeah, that's definitely a good point, that again, the skin physiology changes with later adulthood. So another population to check the reliability on. Great question.
Okay. Okay. Thank you very much for a very interesting presentation.
Right. Thank you, everybody. Thanks for coming. Appreciate it. Have a good afternoon.